Welcome to Behind the White Coat, conversations about science and technology. I'm your host, Carol A. Clark, and I'm pleased to have with me today Dr. Stephen Kingsmore. He is president of the National Center for Genome Resources in Santa Fe, New Mexico. Welcome to the show.
Thanks very much, Carol.
Let's start with your background. I understand that you are from Ireland.
That's correct, yes.
Where were you born?
I was born outside Glasgow in Scotland. I grew up in Northern Ireland, and I immigrated to the States 17 years ago.
And you were originally a physician, I understand?
That's right, yes. I trained as a physician, and then I immigrated to the States, as I say, quite a while ago. Wound up at Duke, spent 6 years there, enjoyed 4 Final Fours and 3 national championships, and received additional training as a doctor. And from there I went on as an academic physician and actually practiced at the University of Florida for a number of years as an attending physician.
And how did you get involved in genome research?
Well, genome research has happened over about the last 15 years, so it's something pretty new. And when I first came to the States, it was right at its start. It was tremendously exciting, and I found myself at Duke, where some of the earliest genome research was going on, and it was a natural place to go. I hadn't done it before in Ireland, but when I got involved in it, I found it to be fascinating and exciting, and I've pretty much stayed there ever since.
For our viewers that might not know, what exactly is a genome?
A genome is the code for pretty much everything a human body or any living organism does. It's a code which you pass on to your kids in heredity, and it encodes every protein in your body, every molecule your body makes, and many of your characteristics, your eye color, your hair color, to a certain degree your height, your weight, whether you'll be good at sports, how smart you'll be. So your genome is really the code of life, and by studying it we can learn an awful lot about diseases, about health, and about so many different aspects of human behavior.
I understand that you actually determined 5 particular diseases.
Yes, so back in the early 90s, the real goal of studying the genome was to figure out the causes of common diseases. And for very many diseases, we don't know the precise cause. At a molecule level, we know that maybe you have a heart attack and we can see the damage done to the heart. We don't know what's the trigger, what's the genetic cause. Did you inherit something that set you up for that? For some diseases, very clearly that's the case. For other diseases, they're complicated, and genetics has a role, but it also has to do with what you eat and whether you smoke and things like that. So for 5 diseases, which were simple diseases, rare diseases, we used genetics to identify the cause of that disease.
What were those 5 diseases?
Well, they've got really big names, but a couple of them were diseases that cause bleeding. And so there's a number of genetic causes of bleeding, and a very well-known one is haemophilia, where people who are born with those genes have a bleeding tendency, and they have to be very careful about, say, contact sports.
They call them bleeders, don't they?
They do, yes. So we studied a group of diseases. One of them is called Hermansky-Pudlak syndrome. And I'm sure you've never heard of it. It's not a common disease, but in certain parts of the world, like Puerto Rico, it's a very common disorder. And we found one of the genetic causes of that. We studied another disease that causes albinism, people with white hair and not much retinal pigment color. And they also have a bleeding tendency. And unfortunately, many of them die young. They die of cancers.
Don't they typically have red eyes?
They do, yes, yes. And so we found the cause of that disease as well. It's called Chediak-Higashi syndrome, after the physicians who first described it. And so that was another one that we worked on. And then an entirely different disorder, one I studied first back at Duke, is called Startle syndrome. And people who have this disorder, and they're quite common in Quebec, they act like they've had 17 cups of coffee. And if you say boo to them, they would literally fall out of this chair. So it's quite similar to having epilepsy, but it's not. It's different, to seizures. and we found the cause of that disease as well, all using genetic approaches.
Interesting that they seem to be confined to geographical areas, Quebec, Puerto Rico.
Yes.
What did you determine about that?
Well, that's part of the makeup of a genetic disease, is it tends to be inherited in a family. And so, especially in a place where you've got some isolation, such as Quebec or Puerto Rico, you tend to have it in a population of individuals. And it can be quite common there, and in the population as a whole, it could be quite rare.
Now, you've determined, for example, the startle disease. What's the next step? What will you do next?
Well, there's a couple of immediate benefits. One is that physicians will then have a test. And so, especially with difficult diseases to diagnose, having a test is critical. It allows the doctor to make a timely diagnosis and then decide on the appropriate therapy. And for some diseases, there could be many, many different causes, and the physician wants to narrow in as quickly as possible, and the patient, to the right one. So having a test is very useful. That test can be used in testing babies to see if they're going to develop the disorder.
Predetermined.
Yes. You know, if that runs in your family, let's say breast cancer, you'd be very keen to know what your individual risk is and what you can do to ameliorate that, to prevent that from happening to you. So for a number of reasons. And then finally, of course, it helps you make better treatments. If you don't understand the cause of a disease, it's really difficult to get an effective treatment. And many diseases today are like that.
What a satisfying field to be in, to be literally changing people's lives for the better and for generations to come. Can we talk a little bit about the National Center for Genome Resources?
Yes.
When it came about, what's the mission and goals?
Yes, the National Center for Genome Resources, I'm the president. I've been there for about a year, and so I moved from the East Coast to sunny New Mexico. We're located in Santa Fe, and we're nestled right at the bottom of the Rocky Mountains, so it's a beautiful place to live. We have a great standard of living, and how we came to exist was that science, and in fact the biological sciences, are undergoing a massive transformation. It used to be that people studied one disease or one gene, the way I did, say, for Startle syndrome, for much of their career. And they would focus in on one molecule. And over the last 15 or 20 years, the way people do science has changed. One of the real leaders in that was Los Alamos National Lab, with starting to sequence the human genome, the entire 3 billion nucleotide code, and to start to study everything at once, all of the molecules in a system simultaneously. That's called systems biology. And in order to do that, you need people who can handle that amount of information and can provide that to scientists all over our nation. And NCGR was created to meet that need. It was a brand new need when we were founded about 11 years ago, and specifically we were sort of an offshoot of Los Alamos National Lab, designed to help scientists here to do exactly that, to do systems biology. Now back then Los Alamos was one of the only places doing that. Now pretty much everywhere across our nation scientists do these type of experiments, looking at thousands of genes or thousands of molecules at a time, and there's a need for them to be able to look at all of the information that's available simultaneously. That's what we do. Were
some of your original researchers 11 years ago actually from the Los Alamos National Laboratory?
Yes, some of them were. Mm-hmm.
Are they still there? Or do they collaborate back and forth?
Yes, we still have pretty good connections with Los Alamos. We focus these days on some different things. The human genome's now sequenced, and that's not happening anymore right here in New Mexico. But we're doing a lot of work together in terms of looking at pathogens and infectious agents. And that's taken a whole new turn recently with the threat of weapons of mass destruction. That's something that Los Alamos is very interested in, and we help provide the same sort of analysis tools, internet-based, web-based tools to help them further their research.
Now, are you involved at all in bioterrorism as it relates to Playas, New Mexico, a new center that was just inaugurated a few months ago that's specifically working on that with the University of New Mexico at Las Cruces?
Yes, we do. We have a lot of interaction with New Mexico Tech. And they're
Who is the owner of Plius?
Right, they're running that facility. As yet, that's just started. That's work that's really just started. We do work with them in the bioterrorism area, but not yet in terms of Plius, and it'll be very interesting to see what happens there. That's a really unique facility.
What are some of the most interesting research your colleagues are doing right now at your institute in Santa Fe?
We're doing a lot of really interesting things. Let me highlight a couple. We do a lot of work related to agriculture and related to making better crops, or crops that are resistant to various diseases. And that's work we've done for quite a while. And we do that collaboratively, so we don't do research just by ourselves. Generally, we work in a consortium with investigators at universities all over our nation. And our role in that is this information gathering and providing it over the internet to scientists all over our nation. They might be at the U.S. Department of Agriculture, or they may be in private industry, or they may be at a university, and they need to connect. They need to be able to communicate with one another, and they need to be able to analyze very large amounts of information, and we assist them with that. So in particular, we work with pathogens that infect a lot of crops, infectious organisms that can really decimate a crop. Back in the history of my people, the Irish people, the potato famine about a century ago, a little bit more, really devastated the population. It caused great hardship, and it led to millions of people immigrating to the east coast of the United States and then filtering across, even as far as Santa Fe.
Through Ellis Island.
Yes, and that came about because of an infectious agent that infected the potato crop. It's called Phytophthora, and we actually study that in collaboration with people at Ohio State University and other institutions. And that's a good example of the work that we do.
I was going to mention, Doctor, that just as the potato crop devastated Ireland, something that would devastate New Mexico would be an infestation in our chili crop, because we are world famous for our chili. Are you working on anything in that realm?
We are, yes. Funny you should mention that. I had really not tasted chili and didn't have an appreciation of this until I came to New Mexico. And now I probably have green chili stew 3 times a week, so I'm one of those people who's a recent convert but thoroughly addicted to green and red chili. And this is a whole new thing for me. But yes, we are working, there's a bug called, again it's a Phytophthora bug, and it infects green chilies. It also infects tomatoes and other vegetable crops all across our nation, all the way from Long Island to California. And it causes billions of dollars of damage in a bad year, a wet year when it's moist. And one of the things that we're just starting to do, we just put in a proposal, is to look at the genome of that particular bug and understand that, and then figure out ways to combat it. There are not good treatments available today to eradicate that, and when a field becomes infiltrated with that particular bug, it's almost impossible to eradicate it. And that's a major limitation to growing things like cucumbers or tomatoes in various parts of the country.
Were you at all involved in last summer's apple maggot infestation, commonly known as the railroad track worm?
Yes, no, we weren't.
Were you aware of that? In northern New Mexico, it was devastating apple crops.
Yes, yes. And we have another bug which is infecting our piñon trees. And then recently our soybean crops have been attacked. So this is a very dynamic thing which happens in our world. The bark beetle. Yes, indeed. There are continually new or emerging pathogens which we're not familiar with. We may not have good treatments for them, and suddenly they become an epidemic and they cause devastation. And part of the work that we do in terms of genetics is to get a basic understanding of these organisms so that when an epidemic occurs, we can diagnose it really quickly and we can treat it effectively. Very much the same that we do with human diseases, with human infections. You know, this winter we've had a lot of talk about influenza, about a new strain that people hadn't expected, and how our vaccine stocks were impaired. And that's been major headline news this winter. And that's an organism that really every year we have to have a new type of vaccine and targeted therapy. And so we continually see this, and this is a very active area of study.
Do you study anything, for example, like Alzheimer's disease, the cause of that? Have you gotten involved in that at all?
No, we're not really involved in that. So we're not a huge research institute, and we have focused in on 3 or 4 areas where we're really good. And we're really good at mathematics and applying that to biological problems. We're really good at software engineering. We're not Microsoft, but we make software for scientists to allow them to analyze their information. And we really focus on infections, plant infections, livestock infections, and human infections. And we also focus on genetics and genomics of genomes of different organisms. Those are 2 of our key areas that we work in.
Have you, for example, the diseases that you were, the 5 that we spoke about earlier,
Yes.
that you determined, once you determine that disease, then have you all started working on cures for those or treatment for those, or does that go to another institution, that piece of
Yes, it really depends. For those particular diseases, I'm not working on them currently. I'd like to get back to them. But what we do like to do is to put together sort of a toolbox for a disease so that researchers all over our nation are well equipped, no matter where they are geographically, to tackle that disease. And then generally they start to investigate the disease in more detail. They've now got the tools, they can really effectively investigate it. And that means better diagnostics or earlier diagnostics and better treatments. And of course better prevention. That's the best thing of all, if you can prevent the disease in the first place. And that takes a lot of study. And we certainly are involved in that. And then what we like to do is to partner with private companies or public companies, biotechnology companies and pharmaceutical companies, and they're the people who make the better therapies. And that's a really good sort of role that we play to foster that translation from going from the toolbox to making some discoveries and then getting them to the public using companies who make products.
I understand that you give talks all over the world, and not long ago gave a talk at Princeton University,
Yes.
Of which I happened to look at a PowerPoint presentation of your talk. Very interesting. Can you tell our audience a little bit about that particular presentation to Princeton?
Yes, sure. This was a talk about better diagnostics, and this is a huge need in medicine. For many diseases, a doctor sees a patient, let's say the patient is short of breath. What's causing that? Well, that person, if they're in an emergency room, they might be having a heart attack. That's an emergency, and they need to have an immediate therapy. Or they could be in heart failure, and that's not quite so serious, but also deserves treatment. And it could be many other things. What the physician needs is a rapid test that will tell him it's this, and then he can treat that effectively. And so my talk was about how genetics is changing the way we diagnose disease. Not just in terms of you've got this disorder, or no, it's great, you don't have that disorder, but also in terms of, and this is the right treatment for you. And what we're learning, and this is something we've only started to realize, is that one medicine isn't good for everybody. Just very recently we've seen a lot of anxiety in patients who have arthritis, because one of the leading medicines to treat people with arthritis had to be withdrawn from the market when it was discovered that a small proportion would have a serious side effect in terms of their heart. And we're starting to understand that that's partly genetic, and that it's possible to make a test to give a physician so that when he sees somebody with arthritis and he's going to pick a therapy and he's got 7 available, he can say, based on your genetics, this is the right treatment for you, and be able to avoid those adverse effects and be able to get you the most effective medicine.
That is so true, because just as a simple example, when somebody has a headache,
yes,
for example, in the office where I work, one particular common over-the-counter medicine will work for one person, and someone will say, I have a headache, and they'll say, I have some such and such, and they'll say, either that doesn't work for me, or that gives me a stomach ache, that hurts my stomach. So that's exactly true. One particular medicine does not work for everybody and has different side effects for different individuals. So what a great thing that is.
We're just starting to pull that apart. We're working a lot with companies who work in this space, who make diagnostic tests. We absolutely need them. But we also need physicians involved, and we need universities to be involved. And our role in this is to sort of manage that information and make sure these guys can talk to each other. They speak a different language, and that we are effective in getting those new diagnostics.
And what else did you present at your Princeton talk?
Well, really, the thing that I focused in on was one specific disease, and it's one that's pretty personal for me. It's called sepsis. And sepsis means a systemic infection. We all have probably 2 or 3 different infections a year. A head cold, something like that. That's an infection. But when that infection moves into your bloodstream, it's called sepsis. And sepsis is a really serious disorder. It kills about 1 in 5 people who have it, and it's the 10th leading cause of death in the United States. Now, I had this a couple of years ago. I was unfortunate and I hurt my back. I needed to have a surgical procedure, and about a week after that, I developed sepsis. And I was miserable. I lost a lot of weight, which was good for me at the time, but I was pretty much out of action for a period of months while the doctors diagnosed me and then treated me. Now, what's needed in sepsis is a way to really pick this up early, and if possible, to figure out, are you going to get much worse? Are you at risk of death? Or are you going to be okay with our antibiotic therapy? And I focused in my talk on how we were studying that in one of these collaborations with drug companies and universities, and our results to date. And it looks like we're going to be able to devise a test in the next 5 or 6 years that will be able to have a doctor immediately within about 15 or 20 minutes go, I think you're going to develop sepsis and we better get you into hospital.
How absolutely fantastic. Think of the lives that will be saved with early diagnosis. Yes. And the misery of not knowing and possibly just getting sicker and sicker, and while they're trying to determine that this person has this situation.
I wish I'd had that available.
Could have saved you a lot of agony.
Yes.
That is wonderful. So maybe in the next 5 or 6 years, this will come about.
Yes, these things are slow. It takes a long time, and you have to be really careful. You have to make sure your test is specific. You don't want to give patients the wrong information, and it has to go through regulatory hurdles to make sure it's safe before it's approved for use by physicians.
So if you were able to, once your research is complete and you find this diagnostic test, for example, if you went into an emergency room feeling very sick, they could give that test and know immediately that you did have it. If you did have it, could begin treating you with the correct antibiotics immediately to try to eradicate it as quickly as possible.
Yes.
How great. Think how many people may have died that it was never diagnosed. No one really knew what caused their death.
Yes.
They just got so sick they died.
Yes, and that's true not just of sepsis. There are many disorders. Take cancers, for example. It's really important to know as early as possible. If you can catch a cancer early, there are a large number of ways to treat that effectively. And if the diagnosis is delayed, the options are rather fewer, and the prognosis, the risk of you dying, is much higher. So in many, many diseases this is the case. We want to move back to early diagnosis. If possible, we'd like to know while you're still healthy. We'd like to know based on your genetic profile.
If you're predisposed.
If you're predisposed. And then you could avoid certain things, and that's really the ultimate goal of the type of research we do. It's going to take a while to get there, but that's our real hope. And that way you can avoid seeing the doctor altogether.
I must say, what you and your colleagues are doing at the National Center for Genome Resources is just, it's changing the world, and how great. I applaud you and your colleagues. We're down to about 2 minutes. In closing, what Is there anything that you would like to say or like the public to know that we haven't covered in the last? We've got about 2 minutes to go.
We've covered a lot of what we do. Let me just think. I think something that we're really excited about right now is really the potential of genetics. And we're starting to think about how we can make this applicable to New Mexico. So one thing I'm keen to do, we haven't started to do it yet, is actually to sequence that green chili we talked about a few minutes ago. And so we're just starting to put together a plan for this right now. And this is something that will be a little bit different for us, but that's something that we're keen to do. And we're starting to get involved in that area. So that's something we really haven't covered, and that's a little bit different for us to start to do some of that genome sequencing ourselves.
And also, I forgot to talk to you a little bit about the widespread allergic reaction to peanuts.
Peanuts we're very involved with. We got into this sort of sideways or backwards, but for a number of years we've been studying a class of plants called legumes. These are beans, and peanuts are part of that family. And it turns out that about 1 in 100 people is allergic to peanuts. And if you've traveled on a plane recently, you'll notice pretzels are in and peanuts are out.
That's correct.
And that's because of that recognition. And what we'd like to do is to understand what makes a peanut give somebody allergy. And then can we breed a peanut that lacks that so that you can have peanuts back on the plane again. I prefer peanuts to pretzels, personally. And so that's an area we're actively working with. We're working with the guys who grow peanuts and produce the peanut strains, as well as the U.S. Department of Agriculture and researchers all over our nation. And part of that will be to understand the genetic sequence of peanut. And it turns out that the peanut genome is really complicated. You wouldn't think so. Little peanut, big genome. And so this is going to be tough, but that's an area we're working on.
Dr. Stephen Kingsmore, thank you so much.
Thank you, Carol.
And thank you for what you and your colleagues are doing for all of us.
Thanks.
And thank you for joining us on Behind the White Coat. We'll see you again next time.